But I had a friend who was lactose intolerant, and if he ate ice cream, oh man, did he get gassy. It would shake the room. I'm really glad that I'm not dead, and it's only because my body can pump out tons of hemoglobin all the time. None of it really makes sense unless you can see it happening. Proteins are all about their shape, and they come in funny shapes and sizes, and they're assembled by amino acids. Let's get into how they get made. Redeem Academy. Hey, what is up, science squirrels? Johnny here with Redeem Academy. I am here to help you get the most out of your education while giving God all the glory he deserves. We are on lesson 8, the final lesson of the DNA unit. It's all about translation, how RNA is used to make protein. 3 things I'll be going over today: why proteins matter, how ribosomes work, and how mRNA and tRNA are used to make proteins. In our notebook, which you can get at redeemacademy.org, if this is your first time watching, we have the big question. And the big question says, in translation, the cell turns a message, a gene, into a protein that does a job. What is a message from the Bible, a verse, that needs to do a job, an action in you? Getting personal. All right, so we have seen how the message from the gene gets out of the nucleus. And now today we're going to see how it creates a protein in translation. But I want to review that a little bit. So let's get into it. So you might remember this. This is our cute little notebook cover. This is transcription, making mRNA. That was transcription. We already saw that. Now here is what we're going to learn today. The ribosome pumping out a protein. All right, so the idea that video was showing us was that the DNA was transcribed by that cute little RNA polymerase into RNA, and then the RNA jumped over to the ribosome and did translation to make a protein. And this is called the central dogma of molecular biology. And that is the big thing happening in cells to make cells do their job of making proteins. Now, if you think long and hard back long ago, we did a cells unit. And in that cells unit, there was a lesson on animal cells, and there was a lesson on plant cells. And in those lessons, there was this tiny little black dot called the ribosome. And I told you about how I went to a toilet paper factory when I worked at a company in college, and there were all these different things I talked about. But the main job of cells, I told you back then, is to make proteins. And that little tiny black dot called the ribosome is going to be the star of the show today. I actually call him the hamburger bun of the cell.
Very fitting, John.
But from far away, he just looks like a little black dot. And his job is to make proteins. And proteins make up a lot of the body or the cells, but they also do the jobs of the cells. And so I actually want to move to a video now to just show you how important proteins are and give you a little more knowledge of proteins, because today is about how the cell makes proteins.
You might already know that proteins are a fundamental part of your diet. But proteins aren't just nutrients you need for building muscles and running around. Proteins are much more interesting than that. However, before explaining what proteins really are and what they are made of, we need to talk briefly about one of my current favorite topics: molecular representation. You see, a protein is a molecule, which might not be evident to most people because molecules are usually represented like this or this.
You remember this ball and stick model? This goes all the way back to the chemistry unit. You got space filling model there and ball and stick model, but then he's showing you another representation of a molecule.
Turns out, however, that this diagram, aptly named a ribbon diagram, is one of the many ways to represent proteins. And look, initially I wasn't super happy with all of the freaking ways to represent the same exact thing.
Whoa, rage monster. So angry about the shapes of molecules.
But when you consider that humans consistently use several representations for the same concept, and that proteins are so small they are literally invisible, or at least they challenge what visible is, then you start to understand the need for different models. Each model captures only some of the features of the molecule.
Okay, so this is huge. So notice that he's telling you that there's 4 different ways to show a protein. He's showing you the exact same protein 4 different ways. Now, you're already familiar with ball and stick and space filling model if you've paid attention to me. But ball and stick and space filling are not the way that you normally see a protein. You usually see what he's calling molecular surface or ribbon diagrams when they show you a protein. And the molecular surface is how they show you the actual shape of a protein, and the ribbon diagram is how they show you the real structure of a protein.
But in the end, they are all simplifications. In a ribbon diagram, you don't see individual atoms or the surface of the protein. However, you can see how a protein is a single chain that folds into a specific structure. The path of the chain is where the backbone of the protein is located. Side chains are attached to the backbone, but they aren't displayed in the ribbon diagram because that would obscure the structure. If you took the backbone, unfolded it, and labeled the different atoms in it, you'd quickly identify a repeating pattern. That's because the chain is actually a sequence of special molecules called amino acids, all of which share a common set of atoms that make up the backbone. There are 20 different amino acids, each with a side chain that has a different shape and different properties.
Now remember, those amino acids correspond to the activity we did with the MERPs. In that activity, you had to use the genetic code, and those 20 things up here are the 20 different amino acids that exist that make up all the different proteins that the body makes.
Thousands of amino acids can combine to form a single chain, which, depending on its length and the order in which the amino acids are clicked together, can fold into different and unique structures. This folding happens because in the molecular world, everything is constantly jiggling, which exposes different amino acids on the chain to each other, allowing them to interact by attraction and repulsion, just like magnets.
Oh my gosh, that's so huge. So proteins are always chains, and they're always jiggling. I love how he says that. They're always jiggling, and they fold up into a particular shape. And with humans, we don't like to judge things by their outward appearance. If somebody just looks at the way you look on the outside, and that's how they judge you, that's kind of what starts to happen in like middle school. Like middle schoolers are the absolute worst for that. Like they just judge you on the way you look. But that's not right, because your soul is what really counts. Your character of who you are as a person is what really counts. With molecules, your shape is who you are. That's all there is to it. Molecules don't have souls. And so the shape of a molecule is what that molecule is capable of doing. And so it's all about what your shape is on the outside. Whereas the shape of a person's face doesn't really tell you that much about a person, but the shape of a molecule tells you everything there is to know about a molecule. When it comes to molecules being totally superficial, perfect.
Reactions can make the chain fold into rigid helical shapes or smooth flat sheets, which can interact with each other too, resulting in a stable configuration, what we know as a protein. And the different properties of the amino acids and the incredible diversity of protein structures are what make it possible for proteins to play central roles in almost every biological process. Some proteins can attach to other proteins to build precise infrastructure. Others will identify and neutralize invading pathogens. Others will function like molecular.
Oh, look at that, ATP synthase. So the shape of ATP synthase is what makes ATP synthase capable of making ATP.
Factories and transform raw materials into finished products.
Boom.
And there are even proteins that can harvest energy from sunlight, which are the ones plants use during photosynthesis.
Which we've learned about chlorophyll. Now, that may not be exactly chlorophyll, that might be a little more than just chlorophyll. Same concept.
Because yes, plants are full of proteins too. In fact, every cell in every living organism is full of proteins, and when an organism eats another, the proteins in the unlucky one will be split back into amino acids, which will then be recycled to create all sorts of new proteins for the lucky organism.
Oh my gosh, what a great example. So what he's telling you here is you eat proteins, break them back down into amino acids, and then reassemble them into the proteins that you need to make.
All sorts of new proteins for the lucky organism, including special proteins that will allow muscles.
Yet another protein I've already told you about, myosin or kinesin, which are motor proteins, also known as John. Okay, is it just me, or does it look like that fox is using some sort of propulsion out of his butt to jump and eat the rabbit?
Of course, protein-rich meals are critical for people too. That's why, with the help of top...
All right, proteins are all about their shape, and they come in funny shapes and sizes, and they're assembled by amino acids. Let's get into how they get made. All right, the process that we're going to be watching today is RNA being used to make a protein. It's called translation, and it's going to happen in the cytoplasm. Okay, now this bizarre looking picture is going to be all the magic. This guy right here, I call him the hamburger bun of the cell. He is a ribosome. Okay, and you're going to see these things flying around. These are tRNAs. So This is a tRNA. And this guy is also a tRNA right here. tRNA. And this one, you'll notice this one has an amino acid on it. Amino acid. So the little circles are amino acids. And so when they have an amino acid, we say that they're charged. Okay? And when they don't have an amino acid, like this guy has no amino acid, we say that they're uncharged. Okay, now there's a lot of moving parts in this picture. I'm going to show you the animation now because none of it really makes sense unless you can see it happening. Okay, so it all starts with this nice girl eating the watermelon again. And so I want to remind you of transcription again. So here's some pieces of watermelon floating around her small intestines. We zoom in on those cells in her intestines. There's the wiffle ball that is her nucleus. And there's a gene. And that's the gene for the enzymes, the proteins for digesting watermelon. And there's the promoter and the terminator for the gene. The gene is on DNA and it codes for making a protein. Okay, there is transcription, there is translation. Now we've already learned transcription. In transcription, you make RNA. Today we're going to learn about translation. It's going to be coming our way. So we're going to skip transcription. So I'm going to zoom ahead. Okay, here's the end of transcription. We see RNA gets made and bloop, there's RNA. Yeah, we made RNA. Okay, so there's our RNA coming out of the wiffle ball that is the nucleus, and it is ready for translation. Now they show you that RNA has nucleotides, A's, T's, no, not T's, A's, U's, C's, and G's. And remember, this codes for something. So remember the genetic code. The A's, C's, U's, and G's, they code for amino acids, right? So it's in translation that we actually see how the coding happens. Remember when we did this on the NERPy MERP? The NERPs! So we discovered that DNA coded for RNA, that's transcription, but then we discovered that GUC codes for a V, and that V coded for valine. Valine is an amino acid. And then if you get enough amino acids, you make a chain of proteins, and a protein causes you to have a certain trait. So that's why they're showing us the genetic code here, because these nucleotides of RNA are going to actually code for something. Now, I didn't put this part in my activity, but there's actually only one codon that can actually start translation, and it's the codon AUG. But there's 3 codons that can stop translation. So just like there's a promoter and a terminator, there's what's called a start codon and a stop codon that start translation and stop translation. And here comes the hamburger bun. Now, for some reason in this animation, they only show the bottom of the hamburger bun, the bottom of the ribosome. And then they show us a tRNA. Now, a tRNA is actually just made out of RNA. Now, this tRNA is charged, so they're showing you it has an amino acid on it. And then the tRNA is going to go grab onto the start codon, waiting, okay? And it grabs on because the codes match. So the codon is the 3 letters that match with the anticodon, and they're like, perfect fit, we're friends. And if it's not the right one, it'll bounce off. But if it's the right one, they bond together and stay stuck together until the right things happen. And then here's the top of the hamburger bun. And then everything's going to start running. Now, I'm just going to let this thing play for a while so you can see it work. Maybe I'll keep talking a little bit. And then there's 3 sites where things come in. There's an anticodon and a codon again. Come on. Still got amino acids, so it's called charged. Yep, okay. Try not to talk too much. Oh, the amino acids connected and formed a protein. And then the E site is where the exit happens. And the A site is where they accept new tRNAs. And it boom, 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 starts making a protein. And this kills me about this video. It is so key for students to see that amino acids are bonding together over and over again. And they just skip it. Like, oh yeah, it made a protein. Like, we could show that a little more, guys. We could let the kids see the process a little longer, okay? But eventually, the reason there's a stop codon is there's this other thing that comes in called a release factor, and it causes everything to fall apart and the protein floats away. Voila. And behold, a protein. And like we learned about, proteins fold up into a funny shape, and their shape is what they do. And now check this out. Remember, this was about a girl eating watermelon. And so it goes from the rough ER to the Golgi, out of the cell. I'm just too fast for this video. Out of the cell, and then those things go and digest the watermelon. And now she has a nice time with the watermelon and doesn't get all gassy. Because I had a friend, now this wasn't about watermelon, but I had a friend who was lactose intolerant, and if he ate ice cream, oh man, did he get gassy. It would shake the room. And that was because he didn't have the enzymes to digest the sugars in milk. And so things didn't go so well. You better hope you got the enzymes to digest that watermelon if you're eating watermelon. All right, let's write down a few words. So the enzyme complex, it's called a complex because it has a top bun and a bottom bun, that reads the mRNA and places the correct amino acid in the growing protein chain is called a ribosome. Go over, well, we got a couple other players that you already know the name of probably. We have the mRNA that contains the message. We have the tRNA, that is the transfer RNA. Now look at that, I just wrote a lowercase letter. You didn't think it was possible, did you? Okay, and if the tRNA has an amino acid, it's charged, okay? And if the tRNA doesn't have an amino acid, it's uncharged. And there's actually, little known fact, ribosomal RNA, rRNA. Because the hamburger bun actually is made of protein and RNA. It's a giant complex of stuff that's really good at causing everything to come together and make those amino acids come together and build a growing chain that is a protein. As these tRNAs fly in, they're accepted by the ribosome into what's called the A site. That is the acceptor site. And then they get pushed along into the processing site. That's what I think the P stands for. You might want to fact check me on that. And then when they're done adding to the chain and they let their amino acid add to the chain, they go over here to the exit site. And so it's just a process of the tRNAs come in, and then they go from here to here, and then they go from here to here, and then they leave. And eventually, a new amino acid will be added to them again, and they can come over here and be part of the process all over again. And I'm sure it's not this organized, okay? And as they build amino acids into a chain, we call this a protein. And this is the magic of how cells build proteins. But here's the catch. They can only build proteins according to the code that is in the message from the gene, the mRNA. So let's say this codes for CGU because RNA has U's in it. The tRNAs are actually just made out of RNA. So we show this little chunk here, but it's really a string of RNA. So we could draw this as a stringy. And so it's a big string of RNA folded up. That's what tRNA actually is. And so right here, this would be GCA, okay? And so this is called, down here, the 3 letters is called a codon, and then the other 3 letters is called an anticodon. And this is a magic code that God created that works perfectly to make the right protein every time. Because if you have the wrong letters for your protein here, then you build the wrong protein, it has the wrong shape, and it does the wrong job. But if this says CGU right here, then we could bring this guy over. Say, oh, if this says CGU right here, then we could go here and we could say CGU. Then it will code for the amino acid R. And this is this right here, which means this amino acid would be the amino acid R. And then we could look right here and we could say, well, what is the amino acid R? R is arginine. So this code is coding for a very specific amino acid right here. And you have to put the right amino acids in the right places, or else the protein has the wrong shape, and then the protein doesn't do its job right, and then the person gets a disease and their body doesn't do the right thing. Now, not all diseases are caused by bad shaped proteins, but a lot of genetic diseases are caused by proteins that have the wrong shape. And there are some terrible, terrible, horribly sad diseases where one amino acid is the wrong amino acid, causes a protein to have the wrong shape, and it's just a horrible disease because of it. This is something you'll see some real examples of in our next unit.
I will see you there, sir.
Okay, I want to review this again because it is fairly complex, and I think a separate animation is a good idea. So we're going to zoom in on the nucleus of a cell again. We're going to watch the whole thing again. So this is everything you've learned in the DNA unit, okay? There is a chromosome, there's the DNA coming out, histones, the gene, okay? Chromatin fiber in the background. Here is RNA polymerase. RNA polymerase unzips the gene and is building RNA, okay? And so we're building RNA. You can see it's just a little bit darker than DNA. And it's going to snip the RNA because there's some stuff going on called processing. It's a little more than just processing, but I'm not telling you about it. And now we have the gene and, the mRNA. And now the mRNA runs through the ribosome, and I like this one because their tRNA looks more like RNA, has an amino acid on the tip, and it's also kind of shaped like a T, which is good for transfer RNA. And the tRNA comes in and starts ticking along, and you see the growing protein chain, and the tRNA's ticking in, and out comes the protein. The protein folds into a shape that looks more globular, which a lot of proteins are, and that's it. Now, just like I was going off about the speed of transcription, translation is usually 5 to 10 amino acids per second. So again, God built a system that is incredible. 5 seconds, I think I might be able to clap 5 times in a second. I don't know if it's quite that fast, but 5 amino acids a second getting placed in the correct order inside of a ribosome. That's how fast your body can pump out proteins. This is an amazing system made by God, and it pumps out functional proteins all the time. One of the key proteins our body has to make all the time is hemoglobin, which I've already told you about, and you have trillions and trillions of molecules of hemoglobin in your blood, making your blood red all the time, absorbing oxygen so you're not dead.
D-E-D, dead.
I'm really glad that I'm not dead, and it's only because my body can pump out tons of hemoglobin all the time. This is an amazing system created by God, and there's 100,000 different proteins I think that the body makes. Amazing. Back to the notes. A chain of amino acids is a protein. 3 nucleotides that code for a particular amino acid is a codon. And the 3 nucleotides on the bottom of the tRNA, so this is the codon, this is the anticodon. I'll tell you, transcription and translation is one of my favorite things to teach because you get to look at God's thoughts after him. The design, the complexity, and yet the elegance to the system. Just amazing. God, you are amazing. Big question. In translation, the cell turns a message into a protein that does a job. What is a message that needs to do a job in you? Isn't that amazing? God has a whole encyclopedia of information in every cell of your body, and he only turns on the ones that need to be used in that cell. Your muscle cells, it's primarily myosin, right? It's primarily those motor proteins and a host of other proteins so that the muscles can pull. Remember, muscles can only pull, okay? Your intestines, they're probably secreting some things so that they can break down stuff and then absorb, right? Only those genes are turned on. In the same way, when you read the Bible, the information in the Bible, only particular portions that day are needed for you. What is something going on in your life right now that there's a message in the Bible in there that's that message needs to do some work in you today? I've got one I want to share with you. All right, I watched the last video back. At the end, sometimes you say something, you're like, oh, that did not come out the way I wanted it. I mentioned in the last video that we don't have to memorize the Bible anymore. And I was like, oh, that does not sound the way I wanted it to sound.
That's not what I meant.
What I was trying to say in that was, we have the Bible now. I don't have my Bible here with me. We have the Bible now. Everybody, if you're an American or if you're not in a third world country, like, you have a copy of it, okay? And I was thinking about people in the first century when the Bible was written. They didn't have a copy of the Bible. They literally needed to memorize it in order to know it. They had to memorize it, and then they had to meditate on it. We live in such a privileged time that we just can have a copy of it. It costs almost nothing. Or it is still a really good thing to be memorizing the Bible. Our kids do Awana, or they did Awana when they were little. Our church has Awana. So good to memorize the Bible. But my point with that was, we're so privileged to be able to read the Bible every day independently. That being said, I have been meditating on these verses with some stuff going on in the life of our church. And so some guys and I have been texting about some things, and this has been one of the verses in our minds. Who is wise and understanding among you? Let him show wisdom through works done in meekness. Meekness is not being weak. It kind of almost sounds like weakness, but meekness is strength that's under control. As opposed to someone who has lots of strength and they just don't have any control over it. They're just screaming and yelling. Okay, but if you have bitter envy and self-seeking in your hearts, do not boast and lie against the truth. This wisdom does not descend from above, but is earthly, sensual, and demonic. There's two kinds of wisdom. There's worldly wisdom, and there's heavenly wisdom. For where envy and self-seeking exist, confusion and every evil thing are there. But the wisdom that is from above, heavenly wisdom, is pure, peaceable, gentle, willing to yield. That is the phrase I've been thinking about the most. Willing to yield, full of mercy and good fruits, without favoritism. And the word there was partiality, but I put in parentheses my own paraphrase of that word. Without favoritism and without hypocrisy. I want to have the wisdom from above in the choices I make. And I don't know what's going on in your life, but I highly encourage you to be reading the Bible and meditating on the Bible, memorizing passages from the Bible, so that you can have the Bible working out action in you. Hey, I hope this DNA unit has been awesome for you. It's a really good chance to think God's thoughts after him. Our next unit will be a genetics unit, which is another taste of DNA, but in a more human and personal way. And I think genetics is probably my absolute favorite unit. You're gonna enjoy it, trust me. So come back for the genetics unit. As always, peace out, Girl Scouts.